So thank you everybody for participating in this Pharma's Day. It's the second one that we have organized. The last one was last year. Just to introduce, well, we are now in a very exciting times because the time of trials have begun. We have newly developed or repurposed molecules under test everywhere in the industry, which means that hopefully in a few years we will get something to provide to our families and patients. So today I would like to thank 5 of the pharma involved. We have accepted to participate in this second Pharma's Day. We will have PepGen, Amo, Dyne, Avidity, and Arthex to report their ongoing trial and studies during this morning. In the audience, well, we should have many people. As you see, we have scientists. We have also families, hopefully. We have clinicians, maybe, but you know, clinicians are very busy on a working day as this one. So it means that probably most of these people will have a look to the YouTube channel to look at the video. So the event is currently being recorded, and it will be posted on the Euro-DyMa YouTube channel in a few days. So the meeting agenda, first talk will be in just a few seconds, starting from Arthex, Beatrice Liamuzzi and Judy Walker. Then we'll switch at 9:30 to Avidity with Kelly Ditrapani and Ling Yong Tai. Then Dyne will with Molly White and Stefano Zanotti, AMO with Mike Snap, and PepGen with Michel Medion. And that's all for me, and I would like to welcome now Arthex for the first talk.
Okay, well, thank you. Thank you very much for the introductions and for the invitation, because for us it's a real pleasure to have the opportunity to share our project with all of you, with all of the community, and please give us as much feedback as possible. Well, I will be doing some forward-looking statements. And first of all, I would like to introduce our team. Arthex Biotech, it's a company, in fact, it's a spin-off company of the University of Valencia in Spain. We created the company, I co-founded together with Ruben Artero, a researcher very well known in the field of myotonic dystrophy, today with us also among all of you. And here you can see the heads of each of the departments at Arthex. Judy Walker, our Chief Medical Officer, is here today with me, so she will explain you later the part of the clinical trial design. And well, we are still growing, of course, because we are a small company, but keeping the team lean but effective. Our mission is to treat unmet medical needs, so diseases that have no treatment. And the way we are doing it is by controlling the levels of microRNA in order to modify gene expression. MicroRNAs are small RNA molecules that are the modulators of the expression of hundreds of proteins. In fact, the microRNAs are very well known because what they do is to modulate the expression of proteins that are involved in the same pathway. So the good thing about them is that we can target them in a very specific way. So we can use what we call anti-mir molecules to block these microRNAs and affect all their targets. So by just modifying one microRNA, we could have an impact in a whole pathway. So they are especially helpful for diseases which have complex mechanisms of actions and with several pathways affected. We focused on myotonic dystrophy because by historical reasons, we have been in the laboratory in Valencia. We have been working with this disease for more than myself. I was working more than 12 years. Ruben was working even more time with this disease. And we studied a lot the mechanism of actions of this disease and decided that MIR23B was a good target for the disease. And we are inhibiting this microRNA using a drug that we called ATX01. This is a single-stranded RNA molecule linked to a hydrophobic moiety. That means that we are using RNA that now it's quite famous because of the COVID vaccines. And with this, we are modulating the expression of MIR23B. Hopefully, we will be in the clinics next year. So our plan is to submit the IND to the FDA at the end of this year, and then proceed with the European agency and try to open sites also in Europe as soon as possible. Well, as I am the first speaker, I will make a very short introduction, because for sure all of you know probably better than myself what is myotonic dystrophy and what are the symptoms. It is considered a rare neuromuscular disease, but although it is rare and it's an orphan disease, the prevalence is still quite high. It's an average of 3,000, 8,000. In fact, more than 100,000 patients already diagnosed only between Europe and US, and it is in fact considered a very underdiagnosed disease. There is adult and pediatric population, a very heterogeneous range of symptoms among the patients, but mainly affecting skeletal muscles and also cognitive dysfunction. In our case, we will be first targeting adults or patients with adult onset of the disease in our first clinical trial. Also, I will explain a little bit about the mechanisms, because I'm the first speaker. So I will try to do it quite simple, but the fact is that what the genetic origin of the patient is a mutation inside the gene DMPK. This mutation it's an expansion of a triplet, a CTG triplet, and an expansion means that this region is longer than longer in patients than in healthy population. In fact, when there is more than 50 CTG repeats, the patients start suffering the symptoms, and with longer repeats, the symptoms are more severe, and also the onset of these symptoms appear early in life. However, the symptoms of the patients were not linked to the MPK expression, because the protein that is produced by this gene is normal. The symptoms are due to the lack of function of this protein called MBNL1. Some of these symptoms are directly related to MBNL because they are defects in splicing. So some proteins that are regulated by MBNL are affected by this loss of function. And what is causing the loss of function of MBNL is the presence of the mutated RNA of the MPK. So this expanded region is causing a toxic RNA. And this RNA, it's known to be toxic because it's having a secondary structure that is acting as a sponge for MBNL protein. So MBNL protein gets trapped in this RNA. You can see them, in fact, in this green green foci or green dots. These are MBNL protein inside the nuclei, but they shouldn't be there. They should be dispersed in order to perform their function. So once they are sequestered here, the symptoms appear. In addition, the presence of this toxic RNA is producing the overexpression of the MIR23B. And this microRNA is directly repressing the expression of MBNL. So there are two different mechanisms that are finally producing this loss of function. The sequestration of MBNL, that was a process already well known, and the lack of translation or the repression of the translation of MBNL caused by the upregulation of MIR23B. So all the companies that will be presenting their projects after myself, they are focused on this part of the problem, trying to reduce the MPK. There are other companies trying to interact, to interfere between the protein and the RNA in order to release MBNL, but we decided to focus on MIR23B, and I will explain you why. First of all, MIR23B, it's a very or highly conserved microRNA. You can see that we are targeting this region of MIR23B, which is in orange. And this is completely the same sequence in all these species, from mice, rats, mini pigs, even to humans. So this conservation allows us to use exactly the same molecule from the concept in addition, miR-23B is upregulated in the disease condition in the muscles of the patients. You can see here in gray, we are quantifying miR-23B in DM1 patients and in controls in white. So you can see that it's consistently upregulated in muscles from patients. Also in the mice model, in gastrocnemius and quadriceps, we have upregulation of miR-23B. And these are peripheral derived myoblasts from patients. In fact, these are cells from patients carrying different lengths of repeats, from 1,300 to 300. You can see that the levels of the miR-23B are higher with longer CTG repeats. And as the CTG repeats correlate with the severity of the disease, that means that the microRNA also correlates with the severity of the disease. As I mentioned, miR-23B, it's directly regulating the protein MBNL, which is directly linked to the symptoms in the patients. And in addition, there is already some genetical studies in mice where they depleted completely using genetic engineering, they depleted the expression of miR-23B, and they had a very mild phenotype of glucose intolerance. That will be something very easy to follow in the patients if it happens. So we believe that decreasing the amount of this miR-23B will be safe in the patients. So to reduce miR-23B expressions, we are using ATX01. That is, as I mentioned, it's a molecule that has two parts. The first of all, it's we have an oligo, an RNA molecule that is binding to this miR-23B, which is also RNA. In our case, in the case of ATX01, this is not natural RNA because we included some chemical modifications that are making it a unique molecule that it's stable and has a long-lasting effect. And this molecule, it's linked to a fatty acid. This is a lipid that it's improving the distribution of the molecule. And this lipid is allowing us to reach the muscle, the skeletal muscle, cardiac muscle, and also the nervous system when we use it in vivo. When we add this molecule, it is binding to miR-23B and blocking it. So then we allow the translation of MBNL. So finally, here, for example, in a human DM1 myoblast, what you can see is that when we treat DM1 with ATX01, here in red, we have an upregulation of MBNL. So we can measure that we have like 4-fold more protein in this case. These are in vitro results in cells, but we also have some results in vivo in the mice model called HSALR from the University of Rochester. Here we were performing a single dose injection, intravenous injection of our compound in this mouse, and then quantified the levels of MBNL as a measurement of the target engagement of the therapy in different tissues. As you can see, the highest levels, we obtain them in quadriceps, gastrocnemius, which are skeletal muscles, also in diaphragm, which will be relevant for the respiratory problems of the patients. But we also have a strong upregulation of the protein in heart and also in brain, which are another two tissues very relevant in myotonic dystrophy, especially in the brain, because the oligonucleotides tend to are very difficult to go through the blood-brain barrier. So normally it's very difficult to have an effect in the brain when you inject intravenously. But our molecules are very small, and with this combination of the fatty acid, we allow also reaching the brain. So as I have explained you, adding ATX01, we have a strong upregulation of MBNL. But in addition, what we observed is that decrease of miR23B expression produce a downregulation in DMPK. So we have a second mechanism of action. This is what we call dual target therapeutic approach. We are reducing DMPK, and what we do is to reduce this foci. We are reducing the toxic RNA. Now MBNL is dispersed in the nuclei because we allow the delivery, the release of MBNL from the foci. You can see here the quantification of DMPK foci in these human cells. We reduce like 50% foci, and this is allowing us to release MBNL from foci. So finally, we have an effect in both mechanisms, in the translation of MBNL and also in the reduction of toxic DMPK. And finally, we produce a rescue of the symptoms. Here, this is just an example of some of the experiments we have done in the mice models to show the efficacy of our drug. Here we are using a model which is an adult onset model. You will probably hear later on a lot about these model mice. They are expressing 220 CTG repeats only in the skeletal muscles. So the only phenotypes that we could assess are in the phenotype, in the muscles. And here, this is just as an example, the grip strength of these mice is reduced in comparison to the controls. So here, the disease mice is the gray line, and the control or wild type condition is in blue. And as you can see here, we were giving one intravenous injection every month, every 30 days. And even at the lowest dose, even at 3 mg per kg, we have an almost complete rescue of strength in the mice. The difference between the lower dose and the upper dose of 24 was that with higher doses, what we have is a more a long-lasting effect of the drug, so higher duration of the effect. And in the muscles of these mice, before the sacrifice, we can quantify the levels of MBNL1 as a measurement of the target engagement, and we see a dose response levels of MBNL reaching levels which are even above the control condition or the wild type condition. We can also quantify splicing rescue. This is the percentage of splicing rescue. So considering here 100, the level of the wild type, and 0, the level of the disease mice, we are rescuing about 50% of the splicing alterations in this mice model. Importantly, these mice do not have the mutated DMPK. So in these mice, we only see the effects of MBNL upregulation, and we cannot see the effects from MBNL release. So we are underestimating the potency of our drug in the mice. Finally, what we have seen, we have done a lot of preclinical studies already. What we in rats, mini pigs, and also in non-human primates. We are still analyzing the data of the non-human primates, but the most positive data is that we have never reached toxicity in these animals, although we use concentrations that were like 30 times the efficacious dose, which is very good news. We are using MBNL to measure the target engagement of our therapy, and this is something that we are doing in the rats, in the mini pigs, also in primates, because it's important to see that we are reaching therapeutic levels, and we will be doing it also in the patient biopsies when we go to the clinical trial. What we have seen is that we have a sustained effect, which will allow to give a comfortable doses to the patients, a comfortable treatment. And we have a therapeutic window which is wide enough, even higher than some of the drugs that based on oligonucleotides that have been already approved. As I mentioned, the results in the non-human primates are very positive, and the toxicokinetic profile, it's also well under control. We can measure the level of the drug in the blood and also in the muscles. I will leave now the stage to Judy to explain you more about the clinical trial plans.
Great, thank you, Beatrice, and hello, everyone. So, as Beatrice mentioned, we've done a number of non-clinical studies, and they are looking very, very promising, both in terms of the efficacy in the mouse models as well as the safety. So, we are getting ready to go into a clinical trial, and we hope to start this next year. This will be our first in human clinical trial, and slide is showing you the overall design of the study. It's essentially a 3-part study, and we're looking at starting very safely and cautiously, because we have to be cautious when we go into humans, and we're going to try increasing doses slowly. So we'll start with a dose level that has been determined to be safe based on the results in animal. And then if that goes well, after dosing 8 patients with either the active drug or placebo, then we'll go into dose level 2 and dose level 3 and so on. At some point, once we start moving up the different doses, and if we're starting to see an effect, we will then move into what we call the multiple ascending dose. So in this part 1, it's patients will receive a single dose, but then in the multiple ascending dose, they'll receive 3 doses 2 weeks apart. Patients who are in part 1 will be allowed, if they want, to continue into part 2. So, and then we'll study probably about 3 dose levels. And we'll see which one of these works the best. And then we'll take that dose, maybe 1 or 2 doses, into an open label extension. So the patients who are in part 1 or part 2 will then roll over into an open label extension, during which we are planning to treat the patients for 1 year. Now, if we go to the next slide, one of the questions that we always get asked is, who will we be doing this? Who are the patients we are looking for the study? So when you do a clinical trial, particularly a first one, you want to make sure that you have a fairly homogeneous group to do the study. Now, we know that DM1 is not a homogeneous disease, but we're trying to limit the first study to the patients where we think we can see an effect most quickly. Later studies will explore other groups, so don't panic if you want to be in the study and you don't meet these criteria. But essentially, the first study will be done in adults with DM1 and with classic DM1. Later, we can look at childhood and congenital DM1, and also I'll talk about DM2, because we also may have an effect in DM2. So who are these patients? We're going to enroll patients who have more than 150 repeats in their CTG gene, who are adults, who are able to walk, and who have grip myotonia. So we want patients who are fairly early in the course of the disease. We're going to exclude people who have other conditions that might make it unsafe for them to participate in the study, and including the severe cardiac disease. And we're going to exclude patients who cannot undergo muscle biopsies, because as Beatrice mentioned, muscle biopsy is really important to know if our drug is working. So if you're on a blood thinner and you can't stop it for other reasons, or you've got a problem with your clotting, you won't be able to participate. We also don't want patients who've been on another investigational drug within 3 months, because then we don't know which, if there is an effect, what does it do to? And then finally, mixilotine, we want patients to stop taking that 21 days prior to screening, so that we can evaluate, are we having an effect on myotonia? So these are our draft inclusion-exclusion criteria. I must say we're still working on the study design, so these may change, so don't hold us to it. And if we go to the next slide, please. The question is, what are we going to measure? Oh, no, sorry, go back one more. Yeah, what are we going to be measuring? So we are going to be measuring, firstly, this is a study of safety and tolerability. We want to make sure that the compound is safe. It's very safe in animal, but we need to be cautious going into human. The next thing we're going to do, as Beatrice mentioned, we want to see, do we see in humans what we see in the animals? Are we seeing in the muscles that we are decreasing the miR-23B levels as we expect? Are we seeing in the muscle of humans that we're increasing the MBNL levels as we expect? And are we seeing rescue of the splicing index as we expect and as we have seen in the animals? So we want to confirm that, and also find out at what dose do we see these changes, so we can determine what dose to take forward. But we're also going to look at clinical endpoints. So how is this affecting the patients? Now, given that it's a small study, you know, we may not see major changes, because you need a certain number of patients to be able to see something that really is convincing, but we are still going to measure a number of different things. And what we're going to measure are some outcome measures related to the muscle. So we're going to measure things like 10-meter walk and run, timed up and go. We're going to measure grip strength. We're going to measure forced vital capacity, which is a respiratory test. We're going to measure tongue strength. We're going to look at quantitative muscle testing. We're going to look at hand opening to see, are we affecting the myotonia, etc. But we're also going to look at symptoms, other facets that are not just muscular. So we're going to test cognition. We're going to check eye function and structure. We're going to check the heart. We're going to check metabolic abnormalities, because due to our mechanism of action, we do think we have a chance of affecting these. And of course, we're going to measure quality of life with a number of different questionnaires. And we're going to be working very closely with the Myotonic Dystrophy Clinical Research Network. Can you click again, Beatrice? I think there may be, no, there isn't. So some people ask us, where are we going to be doing the study? And the study is going to be done in the US, in Europe, and in the UK. So we stay tuned, but there's likely going to be a site near most people who are on this call. And then the myotonic dystrophy type 1, as I mentioned, is our first target. But the interesting thing is that our anti-Mir molecule, ATXO1, should also have efficacy in myotonic dystrophy type 2, because in myotonic dystrophy type 2, MBNL is also important. And so if we can increase MBNL in myotonic dystrophy type 2, we might have an effect in that too, with the same anti-Mir drug. And we're also looking at some other indications for newer compounds.
Hi, everybody. My name is Kelly DiTrapani, and I'm the Vice President of Medical Affairs at Avidity Biosciences. And it's my pleasure to be here with you today to share some information about Avidity and our progress in developing a potential treatment for the myotonic dystrophy community. Thank you to Euro-DyMa for inviting our team to be here with you. Next slide, please. We will be making forward-looking statements during our presentation. So for today, on the next slide, our agenda, first, I will share an overview about Avidity, our approach to patient-centric drug development and technology, and then Lee, who is with me, will walk you through our clinical program for AOC 1001, our compound that's currently in development for myotonic dystrophy type 1. Lee and I are just two members of a large team of people at Avidity who are committed to developing treatments for people living with rare neuromuscular diseases. Next slide, please. So today I'm going to be talking to you about our patient-centric drug approach. And our mission at Avidity our Avidity is a biotechnology company based in sunny Southern California. So I am talking to you from California now, so a little bit in the middle of the night here, but good morning to you all. Our offices are in La Jolla, which is a town near San Diego. And at Avidity, our mission is to improve the lives of people affected by diseases with limited therapeutic options, such as myotonic dystrophy, by advancing a new class of RNA-targeted therapeutics. On the next slide, Avidity is currently working on advancing our compound AOC 1001 for the DM1 community. Our technology is innovative and has the potential to unlock a new way to deliver RNA drugs directly to affected cells, like muscle and heart cells, and allows us to develop new treatments for diseases with currently limited therapeutic options. We are applying our technology to develop our compound AOC 1001, which has the potential to address the root cause of DM1. At Avidity, we have a deep commitment to keep patients at the heart of everything we do. Through partnership with the myotonic dystrophy community, our goal is to incorporate the voice of patients in every step of the drug development process. And I'll be highlighting some of those examples later in our talk. We are pleased to share that our MARINA study, a phase 1/2 clinical trial in adults with DM1, is currently ongoing and enrolling in the United States. And Lee will be sharing more details about the trial in just a few moments. On the next slide, I'd like to talk a little bit about the drugs we make at Avidity, how they work, and how we are hoping to use them to treat DM1. Our potential new treatments are called antibody oligonucleotide conjugates, or AOC for short, considering that that's a long and complicated phrase to say. We are first developing AOCs with a goal to treat genetic causes of muscle diseases, and our first AOC is being developed for the potential treatment of DM1. Our drugs are actually made up of two parts. The first part is an antibody, and many of you are familiar with antibodies because of the important role they play in the immune system and fighting infections. Antibodies are actually often commonly used as treatments because they have the unique ability to bind to a specific protein in the body, and they can be made to target almost any protein, including those that are different between one cell type and another. Antibodies are actually very well characterized for safety and are the foundation for over 100 approved drugs, many of which are for chronic conditions. At Avidity, we are using the antibody as a vehicle to deliver our drugs to the muscle and heart cells. The second part of our drug, as I mentioned before, is called an oligonucleotide, or oligo for short. An oligo is a short synthetic strand of DNA or RNA that's made in a lab. Oligos can be powerful drugs because they can change gene expression or how the body makes a protein by targeting RNA. These oligos can cause the body to increase the amount of a specific protein, to decrease the amount of a specific protein, or to change the size of a protein. And there are currently many multiple approved oligo drugs, many of which are disease-modifying treatments. At Avidity, we are using oligos in our AOCs that are created specifically to target the root cause of genetic diseases. The antibody, in our instance, is designed to deliver the oligo into the muscle and heart cells. So here you see, for muscle diseases, the antibody compound of the AOC enables the targeted delivery to muscle and heart cells, and in turn, improves the power of the oligo. The antibody recognizes and sticks to a specific protein on the muscle and heart cells, delivering oligos to change their gene expression. For AOC 1001, the antibody binds to a transporter protein that's called transferrin receptor 1, or TFR1, in the muscle cells to modify gene expression by targeting the RNA. By combining these two drug components, AOCs have the power of oligos to modify gene expression and the ability to target specific cells in the body, like muscle and heart cells. Here you can see our research and development pipeline. We are applying our AOC technology to develop potential treatments for rare muscle diseases, starting with our first program in DM1 and expanding into Duchenne muscular dystrophy, FSHD, and hopefully beyond. On the next slide, here at Avidity, we really do seek input and partnership with multiple key stakeholders in the development of our treatments. It is only through active and ongoing partnership across stakeholders, including organizations like Euro-DyMa and the community of families they support, healthcare providers, health authorities like the FDA and EMA, and coverage providers, that true patient-centric drug development is achieved. These collaborations can help us ensure that patients and their families are put first in decisions in our shared effort of advancing new and meaningful treatment options. We are continuously seeking input from the DM1 community throughout each stage of the drug development process to best address your needs. Collaborating early and often will always be a key component of our process, and we are doing this through listening, partnering, and connecting with families in the community. On the next slide, patient and caregivers' perspectives truly inform our approach to drug development and inspire us as we stay committed to our mission to improve the lives of people impacted by diseases like DM1. We design our clinical trials in partnership with patients and their families to ensure we are meeting their needs and expectations. And finally, we want you to feel heard and supported and know that you have an open line of communication to our team. Avidity is actually supporting a large natural history study who some of you are familiar with in DM1 called NDM1. One of the important initiatives, this is one of the many important initiatives we are supporting. NDM1 is a non-interventional natural history study, meaning that no interventional drug is being evaluated, but instead the goal of the study is to advance the understanding of the disease progression in DM1 patients. NDM1 is designed and run by the Myotonic Dystrophy Clinical Research Network, or the DMCRN, And the NDM1 study is anticipated to enroll approximately 700 people with DM1 at 17 sites in the United States and in Europe. And I know many of you on today's call are participating in NDM1, and we thank you. Your participation is helping inform and advance the development of AOC 1001 and other treatment options in research and development. Your impact and feedback have truly directly impacted our development program for AOC 1001, and I want to share important examples of how your voice has directly been involved in the development program for AOC 1001. We collaborated with individuals from the DM1 community in a panel discussion to gain feedback to inform the design of our first clinical trial to enhance participant experience in the trial. You'll hear from Lee about some of the support initiatives that we've built into the MARINA trial and overall clinical program for DM1 as a direct result of your feedback. Many of you have also been collaborating with us in a concerted effort to develop a new and important assessment called a patient-reported outcome, which will be validated and used in our clinical trials for DM1. This patient-reported outcome assessment, or PRO, is a disease-specific tool that is a direct reflection of your shared experiences and impact of living with DM1 that will help measure how meaningful treatments are in clinical trials. So thank you for helping advance AOC 1001 for the DM1 community. We couldn't do our jobs without you. And now I'd like to turn it over to Lee, who will cover off on AOC 1001 and the MARINA study.
Thank you so much, Kelly. I just want to do a sound check. Can you hear me?
Yes.
Great.
I can.
Thank you. Thanks, Kelly. Hi, my name is Lee Tai, and I'm an Executive Director of Clinical Development at Avidity Biosciences and also the Program Lead for the AOC 1001 DM1 program. And just like Kelly said, we are very appreciative of our opportunity to speak with you today and to hear feedback from you. Today, I'd like to share with you a little bit about how we're applying our AOC technology that Kelly has shared with you toward addressing the DM1, the disease, and for the patient community, as well as telling you about our first clinical study, the MARINA trial. And I have to thank Beatrice for doing such a great job in explaining the disease mechanism, which I know you all know very well. So some of our cartoons, well, I can kind of skip through quite quickly. As you know, this is a genetic disease that we're trying to target. And the target that we're looking for in our case is the DMPK mRNA. And this toxic RNA is the cause of the disease. And the repeat of these toxic RNA are toxic to the cell and impair normal cellular function in many different organs as shown in this slide. Specifically for our drug, we are focusing on the muscular system. And as you can see from the slide, that a substantial amount of this disease, signs and symptoms of this disease are in the muscle tissue, including skeletal, smooth, and heart muscles. And these systems have a lot of the symptoms that's described on this slide, which I know you know very well. And the important point is that for this slide is that our drug that we're developing for DM1 was specifically designed to go to muscle and heart tissue and to reduce the amount of toxic DMPK RNA in the cell. And we hope that by reducing the toxic DMPK RNA level in the muscle and heart, there will be a decrease in some of these signs and symptoms in these systems. So just a little bit more about our drug AOC 1001. The oligo part of it is a type of oligo we call small interfering RNA or siRNA. And it works by harnessing a natural biologic process in the cell called the RNA interference. RNA interference is a mechanism used by our bodies to fight off things like RNA-containing viruses. So we are leveraging this natural process by delivering a synthetic siRNA that targets specifically to the RNA of interest and leads to this target RNA to be chewed up by a protein complex shown here as a PepGen. In the case of AOC 1001, it leads to this chewing up of the mutant DMPK RNA. So to put it together, the way our drug works is by leveraging the antibody, which binds to the TFR1, as Kelly described, which lead to the cells to take up this antibody oligo conjugate. And once it goes into the cell, the oligo is released from this complex and then go into the cell, both in the cytosol and the nucleus. And it recognizes the DMPK mRNA and interacts with it and bring it to the PepGen, which leads to the degradation of this toxic RNA. So I want to show 2 data slides from the experiments we have done in the lab with animal models. This slide shows the result from a study done in monkeys. The monkeys were given a single intravenous infusion of AOC 1001, and we looked to see what happened to the amount of DMPK RNA in the thigh muscles and the calf muscles over time. The horizontal axis showed the time after dosing when the when the muscle is biopsied. So time 0 will be before the dose. And the biopsies were done on week 4, week 8, and week 12 after dosing. The vertical axis showed the amount of DMPK RNA in the muscle that's biopsied. As you can see on this graph, that 4 weeks after a single IV infusion of AOC 1001, the amount of DMPK RNA measured had decreased by 75%. And stayed down out to 12 weeks after a single dose. Based on this data, we're encouraged that the effect of AOC 1001 may last at least 3 months, which would mean in the clinic, it will be given approximately in 3 months interval. The second slide of data shows that a study done also in monkey. Here we look at many different types of muscle tissues, including skeletal muscle, diaphragm, and the heart. In all of these different types of muscles, we saw about 75 to 80% reduction of the DMPK mRNA. So our animal study showed that AOC 1001 was working the way we had intended to work. And these data support moving this program forward into a clinical stage. And based on this data, we were able to initiate a phase 1/2 trial that's currently enrolling adults with DM1 at multiple clinical sites in the US. And this trial is called MARINA. So this is a slide that describes the study design. So first, the main 3 goals of the MARINA study are to evaluate the safety and tolerability of AOC 1001, and to understand the pharmacokinetics, which is a term describing how the body is processing and breaking down AOC 1001, and also to evaluate the ability of AOC 1001 to reduce DMPK RNA in muscle biopsy. This is a placebo-controlled trial, which means that some participants will receive active drug and others will receive placebo. The active drug and placebo will both be given as an IV infusion, and the participants, doctors, and study staff will not know who's receiving active drug and who's receiving placebo. There are 2 parts to the study, part A and B. In part A, participants will have a single IV dose of either AOC 1001 or placebo and will be followed for 6 months after that single dose. After 6-month follow-up, participants can decide to enter an extension trial in which everyone will receive the active drug. In part B of the MARINA trial, there will be 3 different dose levels, and participants will receive, each participant will receive 3 doses of either AOC 1001 or placebo. After treatment and follow-up for a total of 6 months, they can also decide if they want to enter into the extension trial. This trial will enroll 44 participants and between the age of 18 and 65 years old. Participants must have a confirmed genetic diagnosis of DM1 and be able to walk independently for at least 10 meters during their screening clinical visit. This study is ongoing at multiple sites across the US, and the sites were selected based on their deep experience with caring and managing people with DM1. And further information can be found at clinicaltrials.gov. So at Avidity, our clinical trial programs are designed in partnership with patients and their families, as Kelly has mentioned. And that is true for the MARINA trial as well. And we did this to make sure we're meeting the needs and expectations of participants in our trial, as well as minimizing barriers to participation. As part of the direct input we receive from the patient community, we have incorporated some of the following elements to the trial to lessen the burden and to promote a better experience. These include home health care and telemedicine. And that means that for some of the study visits, participants will have an option to have a qualified nurse coming to their home to complete the required study assessments. And there will also be telemedicine visits that helps with the decrease the travel. We're also providing a travel concierge service to help with arrangement and payment for the airfare, lodging, and ground transportation for the study participant and their caregiver. Participants may also be reimbursed for reasonable trial-related expenses such as meals and parking. During the American Academy of Neurology meeting in April of this year, Dr. Johnson, as the lead investigator for the MARINA study, shared an update on the study progress, which is shown on this slide. The first patient study participant received an infusion in the MARINA study in October of 2021. And also the Part A single dose cohort has been fully enrolled and dosing is complete. From this cohort, there were no serious adverse events observed, and all adverse events were mild to moderate in severity. The Part B multiple cohort is enrolling, and the MARINA study is on track for incremental assessment by the end of this year. This slide summarizes some of the key take-home message from this presentation. And at Avidity, we're committed to listening, learning, and partnering with the DM1 community to better understand the needs of the patients and family we're working to serve and bring forth meaningful treatment options. And our first clinical trial, MARINA, with AOC 1001 is ongoing and currently enrolling patients across the US, and we look forward to sharing an update from the MARINA trial by the end of the year. And also, Avidity is partnering with the global DM advocacy community to provide you with more information and resources as soon as they're available. So I'll close this presentation with this picture of some of the Avidity team members. They are the faces of the company working to address the unmet needs of patients. And we are very grateful for our collaboration with the myotonic dystrophy global community so we can better serve this community and bring forth meaningful treatment options. You can also connect with us at patients@aviditybio.com. So thank you so much for listening.
Whenever you want me to.
Just ask. Okay, you succeeded. Great. And we see your pointer. It's perfect.
That's great. Well, thank you, Lan. Good morning, everyone. It is a great pleasure to be here with you today. I will be making a few forward-looking statements, so please refer to our SEC filings for further information. Here at Dyne, it all starts with our mission. We're extremely focused on delivering life-transforming therapies to people with serious muscle diseases, and myotonic dystrophy type 1 is a priority for us. Over the past few years, we have been validating our technology and its ability to deliver potential therapeutics to muscle. We have generated exciting preclinical data in disease models across multiple indications, and my focus today is to share our comprehensive preclinical data from the DM1 program, and to provide information on our plans to launch our first clinical trial in DM1. We have built a portfolio focused on rare muscle diseases. DM1 and FSHD represent opportunities to develop first-in-class therapies. And we are building a global DMD franchise with exon 51 as our lead program. Apologies. Okay. We are building Dyne as the leading muscle disease company to deliver for families living with DM1 and other muscle diseases. And here you have the 4 core strengths that help us deliver on that commitment. I have a comment from Darren on the chat.
I was just replying to someone else who said they couldn't see the slides.
Yeah,
I can
yeah.
see them.
Okay.
It's okay.
I mean, is there anything you want me to do? No? Keep going?
Nothing you
Okay.
can.
All right. I apologize for whoever can't see the slides. Okay, well, first, we intentionally designed our FORCE platform to overcome the current limitations of oligonucleotide delivery to muscle. The goal for our DM1 program is to develop a therapy that will lead to reduction in toxic nuclear NPK, correct spliceopathy, and enable relatively infrequent dosing to potentially stop or reverse disease progression. Our second core pillar is the focus on rare muscle diseases. We are advancing programs in multiple muscle conditions, and we have generated very exciting preclinical data that really shows the power of our technology across multiple diseases. Our third core strength is our commitment to deliver for people with serious muscle diseases. We are pushing 3 programs to the clinic and plan to be dosing in the DM1 clinical trial participants in the middle of 2022. And finally, Dyne has an exceptional in-house and advisory expertise with deep knowledge of DM1 and oligonucleotide therapeutics. We have a world-class scientific advisory board, which includes luminaries such as Dr. Charles Thornton. As you all know, DM1 is a severe autosomal dominant neuromuscular disease caused by the expansion of CTG triplets in the DMPK gene. The mutations cause the toxic DMPK RNA to form large structures that get stuck into the nucleus and bind to proteins that regulate RNA splicing. This leads to abnormal splicing and ultimately the symptoms of DM1. DM1 presents with myotonia, muscle weakness, as well as cardiac and pulmonary abnormalities. This disease affects tens of thousands of people in the US and Europe. Unfortunately, there are no approved disease-modifying treatments for DM1. Current approaches target individual symptoms, but do not address the root cause of the disease, which is the toxic DMPK RNA that accumulates in the nucleus. There are treatments in development that target DMPK with an antisense oligonucleotide. I'm admitting somebody into the chat. Wait a second. Okay. Note that I will be talking extensively about antisense oligonucleotides, and since it's quite difficult to pronounce for me, I will be referring to them as ASOs. Now, the efficacy of the ASO is limited by poor muscle delivery. This is where Dyne wants to make a difference, and we want to change the way that oligonucleotides are delivered to make a meaningful difference for patients living with DM1. We have developed the FORCE platform to overcome the limitations of oligonucleotide delivery to muscle. The platform is comprised of 3 key components: a Fab, a linker, and an ASO payload. The Fab is a proprietary antibody fragment that specifically binds to the human transferrin receptor type 1. And this is an essential component to enable muscle delivery of a therapeutic payload. This is because the transferrin receptor is expressed on muscle cells. The linker is a val-cit linker that is used to connect the Fab to the payload. This linker is used in multiple approved drugs, and it is broken down after being taken up by the muscle cell to enable release of the ASO payload into the cell. Now, the Fab and the linker are common across our current programs, but FORCE is a flexible platform to deploy different types of therapeutic payloads. And the payloads are rationally selected to match the therapeutic agent to the target biology. And this is different for each disease indication. For example, we've chosen an ASO payload for nuclear targets, such as the one that drive the M1 pathology, or siRNA payloads for cytoplasmic targets. And we really selected a gapmer ASO for the M1 for its known ability to target nuclear RNA via an RNase H-mediated mechanism. Now, together, the Fab, the linker, and the ASO conjugate are designed to target the transferrin receptor type 1, which is expressed here in this diagram at the surface of a muscle cell, for example. And this is really with the intent to harness the biology of the TFR1 receptor to efficiently target skeletal and muscle cells. The drawing here shows how FORCE is intended to work for a disease like myotonic dystrophy. The Fab binds to the transferrin receptor type 1, which is represented by this little gray box here, and the conjugate is internalized or taken up by the cell. In the endosome, which is a subcompartment within the cell that the cell uses to collect material from the outside, the linker is cleaved or degraded, and the ASO is released, first within the endosome and then within the cell. The free ASO then moves into the nucleus and induces the degradation of the mutant DMPK RNA. This leads to the release of muscleblind and correction of splicing. And this mechanism is predicted to stop or hopefully reverse disease progression. FORCE targets the toxic DMPK RNA that is trapped in the nucleus. And in the following slides, I will review with you the preclinical efficacy and the safety of our DM1 clinical candidate, which we have called DYNE-101. We have explored the potential of DYNE-101 as a treatment for myotonic dystrophy in multiple in vitro and in vivo models. We have used cynomolgus monkeys and the HTFR1 DMSXL mice that I will explain in more detail in the subsequent slides to establish the relationship between DYNE-101 concentration in tissue and its biological effect against the DMPK target. And let me thank here Dr. Gurdon for providing the DMSXL mice before we get to the end of the talk. I know she's on the line. We have also used another model, the HSALR mice, to really understand the relationship between splicing and myotonia. And here I would like to thank Dr. Thornton for providing the mice and really helping us with this work. Now, for this model, we have not used DYNE-101 to generate the data you will see, but we have developed a specific molecule that we call a surrogate to really target the disease in these mouse. And this surrogate really mirrors the mechanism of action of DYNE-101. And finally, we have also used cells from patients to really show the potential of DYNE-101 to affect the biology of the disease in a human system. Now, before we dive into the in vitro data, I want to show you this graph. It's a publication from 2017 showing the distribution of CTG repeat lengths in multiple DM1 populations. And these blue boxes here show you where the cell lines that we've used in our program sit or correlate with the level of CTG expansion in the patient population. We have two patient cell lines, one with 380 CTG repeats that really capture the lower end of the spectrum, the least severe forms of the disorder, and another line with 2600 CTG repeats that is representative of the more severe forms, really to test if DYNE-101 can affect disease biology under most of the conditions that will be encountered in the clinic. And this is the data. So on the left, you have the effect of Dyne-101 on the DMPK RNA. In the middle, you have representative images of muscle cells in a dish. Here you have the nucleus in blue. The red dots are the DMPK foci. And on the right, you have the effect of Dyne-101 on the inclusion of exon 11 into the BIN1 RNA. This is a splicing defect that's common across patients and is really a way for us to test whether the effects of Dyne-101 on DMPK result into an improvement in the splicing phenotype. On top, you have the results from the cells with a low number of repeats, and at the bottom, you have the effect of Dyne-101 on cells with a high number of repeats. And you see that in both cell lines, Dyne-101 leads to a reduction in DMPK RNA and also a reduction in the foci. Here you have the vehicle. Oh, there's a question on the line.
Well, we should run the questions at the end of the talk.
Yeah, okay. I don't want to ignore anybody. 1 second. In the middle, you have representative images of the DMPK foci being reduced in both cell lines. And finally, on the right, you see that Dyne-101, upon suppression of DMPK, leads to an increase in BIN1 exon 11 inclusion. And this really speaks to the ability of this molecule to correct spliceopathy. Beyond the in vitro work, in close collaboration with Dr. Thornton, we have also evaluated force in a well-validated DM1 model that is called HSALR. Now, again, remember that Dyne-101 will not work here, so a different force conjugate that mirrors the mechanism of action of Dyne-101 was used, and that's represented as indicated as force. The HSALR mice have toxic repeats in the nucleus and mimic the RNA missplicing and the myotonia that's observed in patients. And this is one of the few models that can model both manifestations of the disease. Now, on the left, you have the effect of force on splicing. You see that there is a dose-dependent reduction in the spliceopathy to the point that in some tissues like the gastrocnemius, the mice treated with force are almost indistinguishable from wild-type mice that are represented here by the green line. And force also led to an almost complete correction of myotonia. And this is the effect of a single dose after 14 days. And you can appreciate by comparing the blue bars to the red bars that are representing the effect of an unconjugated ASO or naked ASO. The force with a single dose at 14 days is far superior to an unconjugated ASO. Now, Dyne-101 is designed for use in humans. So to test its efficacy in a mouse model of DM1, we took the DMSXL mice provided by Dr. Gurdon. These mice expressed a human DMPK RNA with 1,000 CUG repeats. And we crossed them with a mouse that expresses the human transferrin receptor type 1. The resulting combination of the two models allows us to test Dyne-101 against a toxic human DMPK RNA via a human TFR1 mediated uptake. Really, we are trying to mimic what the intended mechanism of action of Dyne-101 will be into humans. In this new model, we wanted to show two things. First, we wanted to confirm that the human DMPK is really, indeed, trapped within the nucleus. And second, we wanted to show that Dyne-101 degrades this human DMPK RNA. Now here we separated the nucleus and the cytoplasm with biochemistry methods from skeletal muscle, from the HTFR1 DMSXL mice. And as you can see from this graph, the DMPK is enriched in the nucleus. And this is demonstrated by the fact that the DMPK expression is consistent with the expression of MALAT1, which is an RNA that's expressed in the nucleus and is commonly used as a control for expression in the nuclear fraction. It's also important to notice that DMPK expression is different from GAPDH, which is an RNA that's usually evenly distributed between the nucleus and the cytoplasm. Here, you have the effect of Dyne-101 in the same system after one dose and after 1 month against the toxic DMPK RNA. In black, you have the control mice. In blue, you have the mice treated with Dyne-101. And here you have the two fractions. And you can see how Dyne-101 really substantially reduces the levels of the toxic DMPK RNA within the nuclear fraction, confirming that Dyne-101 can target the DMPK RNA that is trapped within the nucleus. Now, the mouse, the HTFR1 DMSXL mouse, comes in two different flavors. The data you've seen up to this point was generated in what we call the hemizygous mice. These mice express the human DMPK, but only from one copy of the gene. So there is no splicing phenotype. If you generate another mouse model that with 2 copies of the DMPK gene, you can still monitor DMPK expression, but you also now can see the consequence of this expression as a splicing defect. And you can also monitor the foci that you've seen in the cells. Now we can also do that in vivo. And here you have the results from a 1-month-long study where we tested Dyne-101 in this homozygous version of the HTFR1 DMSXL mice. Here you have the data in the heart. On the left, you see the effect of Dyne-101 on the toxic human DMPK RNA. You see there is about a 50% suppression. In the middle, you have representative images from the heart of these mice showing the toxic DMPK RNA that is stuck here in the nucleus in the control mice and is greatly reduced upon treatment. This is the images on the right. If you analyze by image analysis approaches these pictures, you see that there is a reduction in foci that's very consistent with the RNA reduction, which is again about 50% of a decline in DMPK foci. And now these effects are accompanied by a partial correction of splicing in the heart tissue. This is the data on the right. Now, to guide you through this data, the gray circle is is a control mouse. This is a mouse that does not express the human DMPK. The black circle up here shows you the effect of the mutant DMPK expression on splicing. So this is the difference between healthy and disease. And this is what Dyne-101 is able to do. It's able to reduce the splicing defect in this tissue. Then we proceeded to analyze the effect of Dyne-101 in the same study in skeletal muscle. We have diaphragm and tibialis anterior and gastrocnemius. Results very similar to the heart, about 50% suppression of the DMPK expression, and also a partial to almost complete correction in splicing index in all the muscles analyzed. Now, the data we've seen so far tested the therapeutic potential of Dyne-101. But the next step for our preclinical program was to understand the effect of a monthly dosing regimen. Here we compared a single dose of Dyne-101 at 1 month with 4 monthly doses. We delivered with each dose 5 milligram per kilogram of ASO payload, so a relatively low dose of ASO. And Dyne-101 was able to achieve enhanced human DMPK knockdown in skeletal and cardiac muscle upon low and infrequent monthly dosing. Now we wanted to test whether our observations in mice translate or are reproducible in cynomolgus monkey, in higher species. And to do so, we administered Dyne-101 to cynomolgus monkeys, NHPs. And again, we compared the single dose to 2 monthly doses. In this case, the dose is a bit higher, it's 10 mg per kg of ASO. And again, repeat dosing of Dyne-101 enhances the effect of a single dose. And in this model, we were able to detect up to 70% of suppression in skeletal muscle. You can see here in the gastrocnemius, and a very close level of DMPK reduction in the tibialis anterior. Now, it is important to remember that this is not a disease model. The DMPK RNA is a healthy DMPK RNA, but this is still indicative of the ability of DYNE-101 to target DMPK in skeletal muscle. And to build on this data now, I will show you the complete results from the repeat dose study. Here you have the effects on all the muscles that are, most of the muscles that are critical for DM1 pathology. From the left, you have the heart, then skeletal muscle, and the mesothelial skeletal muscle of the face, and then a sampling of smooth muscle from the GI tract. Here we have the esophagus and the duodenum. Again, there is profound reduction in the DMPK RNA expression, and these data support the potential of repeat monthly dosing of DYNE-101 to drive substantial pharmacological effect in the tissues that matter the most. Now, more importantly, we want to ensure that DYNE-101 is safe. And to do so, we have run a battery of tests, and this is the key experiment that is run, is a 13-week GLP, that stands for Good Laboratory Practices, toxicology study in cynomolgus monkeys. Here, DYNE-101 was well tolerated with no dose-limiting toxicities, up to a maximal feasible dose. There were no changes in cardiac or respiratory or neurological endpoints, no effects on clearance organs such as the kidney or the liver, and no effect on coagulation. And an OAL was identified at the highest dose tested. The data we have reviewed demonstrate the potential of DYNE-101 to target the genetic basis of DM1 and to drive splicing correction in vitro and in vivo. A monthly dosing regimen in mice and cynomolgus monkeys leads to a significant DMPK reduction in different muscle types that are affected by DM1 pathology. And DYNE-101 was well tolerated in a 13-week GLP toxicology study in cynomolgus monkeys. With a surrogate molecule, we have also shown and demonstrated the superior ability of force to correct myotonia in skeletal muscle compared to a naked ASO. These data are the foundation of our preclinical program in DM1, and they build our confidence as we move toward the clinic. Now, to the final part of this talk. We have done extensive work to prepare and advance DYNE-101 to the clinic. We have engaged with stakeholders around the globe as part of this effort. The engagement has included advocacy groups, patients, and key opinion leaders from various disciplines to ensure that they have their crucial perspective on how to inform our efforts. This input is critical and has provided immense benefit to the overall design of the clinical study, our biomarker strategy, our endpoints, including splicing, myotonia, and various strength and functional outcomes. And also key safety considerations were included in this design. Discussions with stakeholders have provided key learnings from natural history studies. We have reached out broadly to advocacy leaders, patients, and caregivers to ensure we have a patient-centric study and provide advice in a number of areas, including clinical trial assessments, placebo and biopsy-related considerations, as well as clinic visit designs and decision-making insights for the selection of participating in our clinical trial. We are also using insights from the END-DM1 natural history study. This was a 2-year-long study, including patients from 18 to 70-year-old, over 700 people enrolled. This is the largest and most comprehensive natural history study conducted to date in DM1, with support for a number of institutions and companies, and we really believe that this input help us design the best possible clinical program as we move forward. Patient, caregivers, clinicians, providers, and regulatory feedback have all shaped our thinking in our proposed study design for DYNE-101. As you can see, we are planning a global, placebo-controlled, multiple ascending dose study for DYNE-101 among individuals with symptomatic myotonic dystrophy type 1. We are aiming to enroll about 40 to 50 adult patients, and the study will also have a long-term extension. As for endpoints, we plan to evaluate the splicing index measure, the muscle strength and function, including myotonia, ambulation, respiratory function, as well as safety and tolerability. We are submitting to multiple regulatory agencies in the second quarter of 2022. And additional critical details, including dose and dose regimen, will be available once we have completed our discussions with regulators. While we are initially focused on evaluating DYNE-101 among patients with adult or later onset disease, we are committed to address the full spectrum of the DM1 patients. So this is a really exciting time at Dyne as we prepare to transition into becoming a clinical stage company. We have 2 co-lead programs. In addition to DM1, we're also working on a DMD program. We've also a 3rd program on FSHD, not far behind. And we are focused on execution on all fronts as we seek to bring our potential therapies to patients as quickly as possible. And now I would like to conclude just with a few comments from the patient community. I'm sure that many of the audience are familiar with such situations. I just give you a few seconds to read the slide, and then I just would like to thank you all for your attention, the people at Dyne that have contributed to our work and will work on the clinical program, and of course Genevieve Gurdon and Charles Thornton for providing the mouse models, and people working at INSERM Paris for providing some of the cells we've used in this work.
Thank you, everybody, for the opportunity to speak today. It's a great pleasure to be in the same virtual meeting with all of you. And I just wanted, if it's helpful, to give an update on where we are with the currently running study in congenital myotonic dystrophy. By way of brief introduction, for those of you who don't know, I apologize if it's repetitious for those of you who do. I am the Chief Scientific Officer of Amo Pharma. We are a small company of 10 people, half in the UK, half in the US. And we set up the company because we have personal interests in treating serious rare conditions that affect child development. So we are testing an experimental investigative medicine called AMO-02 in congenital DM1. We are following the usual process where after phase 1, you do phase 2 studies. Phase 1 is normally done in healthy volunteers. That's what was done here. Then you do an initial exploratory understanding study to decide the parameters that you want to advance in a first patient study. That's called a phase 2 study, and that's been done, and I'll show you some data on that. And then on the basis of that, design a, in this case, single pivotal study, which if it gives signals of efficacy and safety, could be sufficient for approval of the molecule. We did our initial phase 2 in adults with congenital and early childhood onset DM1, and we are now running a phase 2/3 pivotal study, which is coming to the point of the conclusion of enrollment in children with congenital onset DM1. So again, probably telling you things you already know, we sort of think of congenital DM1 as a genetically determined developmental disorder in children. Our background is we have more of an understanding of CNS drug development in these kinds of disorders. So please don't ask me complicated questions about muscle because I might not be able to answer them. And we view the symptoms of congenital DM1 as sort of broadly and crudely could be divided into 2 buckets. One are chronic symptoms that are there pretty much invariantly throughout life and on a day-to-day basis, a lot of which are CNS, so cognitive deficits, signs of autism, somnolence, and so on. And those are things, because they're there all the time, we can measure in clinical trials. But of course, they're not the complete burden for families. The families are of affected individuals are obviously worried about episodic but very serious events that could be cardiac, respiratory, GI, and so on, which are perhaps better monitored post-approval in real-world evidence study or something like that. There's obviously no approved treatment for congenital TM1, and we all know this is a condition caused by the presence of an abnormal RNA that binds to certain key proteins and therefore disturbs splice variation in tissue level effectors. So, what does AMO-02 do? AMO-02 is a traditional low molecular weight pharmaceutical rather than a more larger biological. It is taken once a day orally as a strawberry flavored liquid. Once it's taken in, swallowed by mouth, it will penetrate almost all organ systems of the body. We've measured this in rodents and primates. It gets into smooth and skeletal muscle, to brain, all different levels of organs. And the short version of what it does, and I'm sticking with the short version for virtue of time today, is it reduces levels of the CUG repeat RNA, that is, the pathological pathogenesis of this disorder. We've shown that it will do that in different mouse models, and it will do that in tissue from patients. And again, for reasons of time, I am not showing it, but we've shown that that will correct splice variants downstream of the key proteins like GFK3 beta and MBNL. In transgenic mouse models and in human tissue, and that's all published. And it will rescue histology, overall survival, and tissue function in those mouse models. So that's what it does. As I say, you take it once a day. It should get to all tissues. It should be able to address the underlying biology in all tissues in human beings because it does that in animals. So theoretically, the molecule has the ability to alleviate any of the symptoms of congenital DM1, and that's what we've tried to seek out and test in clinical studies. So I mentioned we've run a small exploratory pilot phase 2 study. The goal of that study was to investigate outcome measures, practice what we were doing, think about the pragmatics of how to run a study, and then to gather safety information, potential signals of efficacy in order to design a pivotal study. This study was run in the UK under the auspices of the UK regulator, the MHRA, and their proposition, which we were happy to accept, is although our interest is in congenital, to first study adolescents and adults with congenital onset. And once we've demonstrated safety and potentially some benefit in that study, then move to a study in children later on. So this study is now published. The reference is there. You can go and get the reference if you want. And this is the study design. It's a specific design that follows a template that we followed before in progressing the investigation of molecules in the clinic in individuals with developmental disorders. So each of the individuals in the trial is stabilized on placebo for a couple of weeks. I'm not showing the data here, but none of the things that we measured over that 2-week period changed during placebo. And then the individuals are transitioned single blind to either a low dose or a high dose of AMO-02 for a couple of weeks, for 12 weeks. They're then taken off drug, and there's a follow-up 2 weeks post the cessation of treatment to see about the maintenance of any benefit that might have been seen. These are 2 groups of 8 patients, so it is a small study. The majority had congenital onset, although a few had early juvenile onset. Their mean age was 22, so adults who had been sick for some decades. We measured safety. We measured a variety of different measures, which you'll see on another slide. I won't go through them in exhaustive detail, but we have a rating scale that's a derivative of work derived from natural history by the University of Rochester and the University of Utah, Professor Chad Heatwell and Professor Nick Johnson. That is a highly validated approach to looking at symptoms across the body in many different domains and organ systems. We also measured the amount of autism in these individuals, something called the Clinical Global Impression of Improvement, which FDA is very keen on, which I'll mention in a moment. We did do some functional outcomes, and then we had some caregiver measures to allow caregivers to assess whether or not the individual that they cared for was improving in relation to things that were important to them as caregivers. And then we are doing biomarker work and measuring the level of drug in the plasma in order to try and potentially correlate that with any safety or efficacy signals that we see. So that was the trial design. Commonly, one uses the Clinical Global Impression of Improvement just to get a general handle in a very first study where we don't know what might be happening or not happening, to see whether we have responders. The Clinical Global Impression of Improvement is a well-validated approach to assessment. The way the system works is you have a 7-point scale indicating change, where a score of 4 indicates no change, a score of 5, 6, or 7 indicates progressive worsening of symptoms, and scores of 3, 2, or 1 indicate progressive betterment of improvement. And we invented a whole rubric of anchors and training to define the difference between a 3 and a 2, or a 5 and a 6 for the investigators to use. And what you can see is out of the 8 patients, the majority respond at the high dose, and pretty much most people respond at the low dose. And I will tell you now, just to save time, the difference between responders and non-responders is predicted by the amount of drug in the bloodstream. So it's not predicted by severity, repeat size, or something like that. I haven't shown it here, but the delay or the time to onset of improvement is about 4 to 6 weeks. So a reasonably rapid improvement in overall function as assessed by this methodology. Now, I'm not going to go through every line and every data point on this slide. There's obviously a lot of information. You can review it at your leisure afterwards if you like, or ask questions at the end of this discussion. But the short version is, we defined in this approach, and we worked with the regulators with this as well as the investigators, a 10% improvement equates to a clinically meaningful betterment in terms of what the individuals were showing. And we showed clinically significant change in most of the rating scale measures that we used. Most of the time, these improvements were dose dependent. Most of the time, there was a very large effect size. That's a metric that we use in clinical studies to determine how well the drug is working, if you like. Where we also showed beneficial changes in walking speed and in myometry in the hand, and some indication of improvement on some of the biomarkers we looked at, although the biomarker work for this study is still ongoing, so there's more data to come on that. So I guess, again, in the context of an initial preliminary pilot study, a rapid onset, broad-based effect that seemed to affect many organ systems. So a promising start and some pointers as to how to design a phase 3 study. These are some of the comments that were data-based during them. This is obviously not systematic data. It's just commentary that was collected during the study. But you can see from this information that those numerical changes in different outcome measures were translating into some improvements in real-world function. So again, a generally positive trend and in an initial small study. And again, I won't go through it in massive depth. These data are published in abstract form at conferences now, working with UCL in London, using their very sophisticated technology to look at the amount of drug in plasma. For all of the key outcome measures, there was a correlation that the more drug that was present in the system, the greater the improvement in the individuals, which again gives a pointer that we can have a little confidence that there's a relationship between the drug and the improvement, which is perhaps indicative that this is a drug-related effect, not just chance or something like that. So on the basis of that, we've designed a phase 3 study that was originally due to start in April of 2020. But as you may be aware, there was an outbreak of a virus that interrupted many processes for a lot of different people at that time. So we had to go on pause, waiting for the pandemic to go down. The study then commenced with specific protections to take account of what might happen during lockdowns and so on. The study started in April of last year in many centres across the world under the control of many regulators. And we've had good relationships with the FDA, MHRA, Health Canada, and the regulators in Australia and New Zealand. And I'll come back to where we are in the study in just a moment. The study is a double-blind, placebo-controlled randomized study, nothing very adventurous there. Half of the individuals are on the high dose of the drug, half of the individuals are on placebo. And during the course of the study, they are given safety assessments that are continuously ongoing, and an independent data safety monitoring committee meets regularly to consider that data. To date, they have not required any significant actions during the conduct of the study after 5 meetings, I think. And then a variety of different measures are being used to assess any benefits that might occur in the study. Similar to the phase 2 study, clinician-completed rating instruments that look at the overall presentation of symptoms. The caregivers are given multiple scales so that they can have their input into what's going on. There are a variety of exploratory measures that assess muscle function, cognition, and so on, and then biomarkers and plasma levels. And the primary outcome measure is something we call the CDM1-RS. So what does the CDM1-RS do? It's trying to measure many different functions across the body, which are listed here. So they go from mobility and movement through autism-type symptoms, communication difficulties, cognition, and other signs. And I'll show you the distribution of scores in a moment. These items are scored 0, from 0 to 4, 0 being the absence of any symptoms, through to 4, the most severe possible and the most interfering. And the scale is administered by the principal investigator at the site on the basis of observation. It's not a systematic interview. FDA has guided us that we should try and look at what the individual can or cannot do. So we come up with a score. The maximum score possible is 44. The average score is about 15 or 16 because this is a pretty variable condition. Not all patients have all symptoms, and I'll come back to that in a moment. And as I mentioned, this is a derivative of work done on the basis of extensive natural history studies, principally at the University of Rochester. So the scale is age independent. We get similar scores for young children or children post-puberty, and it detects the same scores in male and females. So it's quite broad-based. And I mentioned if we look at the individual scales, you can see that the highest scores are for communication difficulties, difficulties in thinking, The next highest scores are to do with movement. And it's an interesting conversation, perhaps for another day, in children with congenital myotonic dystrophy, whether their difficulties in movement are due to muscle issues or whether they are due to CNS control of movement, i.e. whether it's a dyspraxia or a dystrophy. And then you can see that variably other organ systems are engaged. So our point here is we are trying to canvas across all of the system domains. The individual items here correlate with other scores. So for example, difficulty thinking correlates well with formal assessments of IQ. Limitations with mobility or walking correlates well with performance on the 10-meter walk-run test, and so on and so forth. So that's what we're trying to do. Where are we? I mentioned this is a double-blind placebo-controlled study. It involves 5 months of treatment. We have a target of 56 patients. These are children ages 6 to 16. The reason why we picked 6 to 16 as opposed to 2 to 15, doing reliable measurements in our experience in children younger than about 4, 5, or 6 becomes tricky. And then it's a pediatric study, so we define an upper age limit somewhere around 16. The study commenced in April of 2021. We did an interim analysis to make sure that the study was the correct size in February of this year, which concluded that it is. We are closing recruitment down now. We're entering the last few patients. So we anticipate it will have taken us something like 15 months to recruit. And if we close out recruitment in July, August, well then after 5 months treatment, we'll have a readout sometime April or so next year. At the end of the study, individuals have the option, if they would like it, to transition into an open label safety study that runs for 12 months. I think essentially all bar one of individuals to date have transitioned into that. So that is ongoing at the same time and being followed up again by our Data Safety Monitoring Committee. And that is pretty much where we are. It's a molecule that should treat most of the symptoms of the condition by addressing the underlying biology in most organ systems. Initial studies in a small pilot were consistent with some indication of efficacy. So we are now running a study which could be, if it's positive and shows acceptable safety and efficacy, the basis for submission for an approval, and that study should finish in the first half of next year. And that is what I intended to say.
Thank you, everybody, for inviting us to speak with you this morning. My name is Michelle Mellion. I'm the SVP and Head of Clinical Development at PepGen, and we're going to share with you our development program that we refer to as PGN EDO-DM1. I will likely just refer to it during my talk as EDO-DM1. I'm sure that all of you have seen these disclaimers, and these are my disclosures. And during my talk today, what I would like to share with you is what we call our enhanced delivery oligonucleotide technology, and explain that to you, as well as our preclinical data in our HSA-LR mouse, and also share with you our clinical development plan and discuss with you how we would like to continue engaging with the community as well. But before I get into our development program, I just want to take a moment to introduce PepGen to Euro-DyMa. PepGen, for those of you that don't know, is a clinical stage biotechnology company. We are located currently in Cambridge, Massachusetts, and we also have laboratory space in Newton, Massachusetts as well. And what we're committed to is that we are committed to developing treatments for rare diseases. Right now, we are focused on Duchenne muscular dystrophy, as well as myotonic dystrophy type 1. We are approximately 30 people strong and growing. And what we're really trying to do is, again, develop therapies for rare diseases using our enhanced delivery oligonucleotides. And so some of you may be asking yourselves, what are these enhanced delivery oligonucleotides? And I think that from the field, some of you may be aware that while oligonucleotides, and these are the genetic medicines that target the root cause of the disease, and they can be effective, but they're limited because we just simply can't get enough of them to the tissues that are affected to have a meaningful impact on disease. And so PepGen's technology, what we do is that we take these very powerful oligonucleotides and we link them to our proprietary enhancing peptide. And so what we're showing here is PepGen's enhanced delivery peptide. And a peptide is basically a chain of amino acids. And basically, you can see here that there are 3 different domains for our enhanced delivery peptide. And these domains are made up, again, of these amino acids that are separated by this hydrophobic core. And the way that this is designed is to improve exposure as well as tolerability and activity. And so by improving this exposure and putting it together with the therapeutic oligonucleotide, which is the genetic medicine that targets the root cause of the disease, we are able to essentially result in efficient cellular uptake of the oligos in those tissues that are affected by disease, and those tissues that are hard to get to, and especially in myotonic dystrophy, and that includes cardiac, skeletal muscle tissue, as well as the CNS, or the central nervous system. And so our pipeline at this time is committed to developing meaningful candidates for the treatment of Duchenne muscular dystrophy and a myotonic dystrophy type 1. And we also have discovery programs that we are currently evaluating. And so, as you all well know, myotonic dystrophy is a multisystemic disorder affecting skeletal and smooth muscle. And because the smooth muscle is affected, the GI tract is also involved, causing swallowing issues, abdominal pain, sometimes constipation and diarrhea. The respiratory system is also involved, causing resultant from diaphragmatic dysfunction, as well as the cardiovascular system, mainly the electrical function of the cardiovascular system causing arrhythmias. And also there is cognitive dysfunction causing intellectual impairment. There's also a metabolic syndrome that goes along with myotonic dystrophy. And so the point here is that there are multiple organs that are involved in myotonic dystrophy or affected by myotonic dystrophy. And our enhanced delivery peptides can reach most of these body systems and deliver effective therapies, that being the oligonucleotide. And we know and have learned from our program in Duchenne muscular dystrophy that the linking of the peptide to an oligonucleotide results in meaningful exposures in those systems or those organs that are affected by myotonic dystrophy. And here what I'm showing is a graph that was generated, the data was generated in non-human primates. And what we're showing here is that we were actually able to deliver the oligonucleotide successfully, not only to skeletal muscle, but also to the heart, which is difficult to reach, to smooth muscle, that being the gut, the duodenum, which is part of the gut, and the esophagus, as well as to the central nervous system. We saw that there were dose-dependent concentrations. This is on the y-axis. So as we increase the doses, at least for EDO51, we were able to show that there were dose-dependent increases in exposures in these tissues. And we anticipate that it's because of the peptide that we are able to get these exposures. So if we had conjugated the peptide to the EDO-DM1, we anticipate that we would have similar types of exposures. And the theory behind our treatment for myotonic dystrophy is that I know that you all are aware that myotonic dystrophy is caused by a triplet expansion that expands a key protein. And it's that when you have this triplet expansion, essentially you have the formation of this hairpin loop, which acts like a sponge, sopping up critical transcription factors. And one of these critical transcription factors is called MBNL1. And because this is sopped up by this sponge-like hairpin structure, we're not able to process the proteins appropriately. And as a result, these unprocessed proteins cause many symptoms that we had just discussed about in myotonic dystrophy. So with EDO-DM1, what this is really designed to restore transcription factors such as MBNL1 so that they can function appropriately and correct these downstream events, resulting in normally processed proteins and normalization of body functions. And so what our oligonucleotide does is that it essentially binds to this hairpin loop, blocking those sites that are sopping up things like MBNL1. And so MBNL1 is essentially liberated so that it's able to process proteins normally and then do their normal functioning and restoring function to skeletal muscle tissue, as well as to the heart and potentially to the CNS. And we've looked at this in multiple preclinical models, which helps to guide our development planning. We've looked in patient cells as well as in the HSA-LR mouse model, which I believe that you're likely familiar with, but I will describe briefly, and also in normal non-human primates. And so what we've learned from looking at cells from patients with DM1 is that when you look at those cells that are not treated and that have these long repeats, you get these pink dots, which are essentially the foci. And this is where we believe that the MBNL1 and other transcription factors are being sopped up and sort of just being held there by that hairpin loop, and so not able to do their function. And what we did was we treated these cells with the oligonucleotide without the peptide conjugation, and you see some decrease in those foci, but you don't necessarily see what we think is meaningful decrease. But when we take the oligonucleotide and conjugate it or link it up with our peptide, what we do see is a significant reduction in the number of foci in the patient cells. And not only do we see a significant reduction in these pink dots, if you will, and these are the foci in the patient cells, but we also see correction of those downstream functions of those transcripts. And so in this graph, what we're showing is healthy cells here, and then we're showing the DM1 cells in teal. And these are different dosages And we can see here that, I'm sorry, these are different transcripts, rather, apologies. And we can see here that across these different transcripts in these is that there's significant correction of their function. And so when we take this oligonucleotide into an in vivo model, into the mouse, which is called the HSA-LR mouse, and basically what we have seen in the HSA-LR mouse, this mouse was designed to display both the molecular and functional DM1 phenotype. So that's specifically looking at myotonia, that loss of relaxation and muscle contraction. And so how this mouse is modified is that there's a repeat put into the HSA gene, and it causes skeletal muscle specific abnormalities, the sequestration, if you will, of MBNL1 and the downstream missplicing events, as well as the myotonia, that prolonged muscle contraction. And so when we looked in this mouse model, what we saw correction of the abnormalities in those critical transcription factors. And what we're showing here on the horizontal axis is actually the concentration that we have given these mice, and then looking at the correction of the specific missplicing event. So for ATP2A1 and CLCN1, which are involved in cardiac abnormalities with myotonic dystrophy, we see here again that there is this dose-dependent correction of the abnormality caused by those repeats, and so that these transcription factors actually essentially normalize to where this black bar here is those normal mouse to the function of where the normal animals are. Additionally, we not only looked at this transcriptional level, but we also looked at the myotonia, that prolonged muscle relaxation. And so here again on the horizontal axis, we have the increasing concentration of EDO-DM1. And on the y-axis, we measured muscle relaxation. This is what normal muscle relaxation looks like in a normal mouse. And you can see here that there is reversal of the myotonia to normal levels after in essentially a dose-dependent fashion, with at 30 milligrams per kilogram, we actually have complete correction of the myotonia coming back down to those normal levels. And some of you may be asking why we're so interested in myotonia, and clinical studies do indicate that myotonia is correlated with other functions in patients with myotonic dystrophy, specifically fatigue, physical, and also physical function as well. And so we've filmed these animals, and like I said, in the HSA-LR mouse, what you see here on the left is the untreated HSA-LR mouse, and as the investigator here is pulling on the mouse's back legs, you can see that the legs are sort of held in contraction, and that is myotonia. And when we treat the mouse with a single dose of EDO-DM1, what we see is that when the investigator gives the stimulus, is that essentially the myotonia is completely reversed. Additionally, when we took a look at the improvement or the durability of the effect on splicing, what we do see is we see that in those transcription factors, again looking at the ATP2A1 and the CLCN1, is that we looked in two types of striated muscle, that being the gastrocnemius as well as the quadriceps. And we see here that on the horizontal axis is time in weeks, so up to 24 weeks. And this is the measurement of the correction. And you can see here that this is a durable response for both of these transcription factors, lasting up to 6 months. So it's a significant time that there is a reversal, if you will, of the missplicing of these transcription factors. So our plan for EDO-DM1 up through this point, we have been able to show preclinical proof of concept in cells and as well as in animal models. And this year, what we are doing is that we are dosing non-human primates to explore the most tolerable and efficacious dose range. And we hope to also host a community advisory board to evaluate or help us evaluate what a study could look like in patients living or people living with myotonic dystrophy. And we hope to initiate our clinical trial in people living with myotonic dystrophy in the first half rather of 2023. So we will be filing an IND in the United States as well as in other countries. In that case, it would be CTAs. And then once we run the clinical trial, we hope to analyze the data and share that data with the community and with others about the tolerability and splicing data, and also then discuss with you and work with you on the best path forward for development of this potentially efficacious treatment for myotonic dystrophy. And so really it's important to us here at PepGen to have this back and forth dialogue with patient communities such as yourselves to be sure that we understand from you what is important to assess in these clinical trials, and to understand what your preferences are for therapies, as well as to give us an idea of how we can develop meaningful protocols for you, as well as educational materials, and how we can continue to support you not only with treatment, but also with support in clinical trials and just in the community in general. And so you are a key stakeholder in our thinking about our development program for EDO-DM1, and it is important for us to have an open and honest dialogue. And so in summary, where we are right now with our program is that we have been able to show in preclinical studies that we've corrected the molecular and functional phenotype at generally well-tolerated doses. And we've also observed the resolution of myotonia, and we will be initiating a clinical trial in people living with myotonic dystrophy type 1 in the first half of 2023. So I'd like to just thank you all for, again, allowing us to speak with you today. It is truly an honor, and I look forward to taking any questions.